Hi there, everyone. It is time for our next talk. So let me give our introduction. Dr. Alex Wiedenhoeft is a research botanist and team leader in the USDA Forest Product Laboratories Center for Wood Anatomy Research, where his research program emphasizes botanical wood anatomy, forensic wood science, and biocentric wood science. He holds academic positions at the University of Wisconsin-Madison, Purdue University, Sao Paulo State University Batucatu, and Mississippi State University. He's an elected fellow of the International Academy of Wood Science, a deputy executive secretary of the International Association of Wood Anatomists, and an associate editor for Frontiers in Plant Science. His educational activities focus on various aspects of forensic botany, wood science for baseball bat manufacturers, and wood science and technology for the U.S. flooring industry at large. Dr. Wiedenhoeft is one of the pioneers of the Xylotron Wood Identification Program, the inventor of the xylophone, and works actively with law enforcement in the United States and abroad to bring wood forensic technologies to bear in support of legal and sustainable use of forest products. So without further ado, please join me in welcoming Dr. Wiedenhoeft.
Well, thank you, Joseph, and thank you, everyone, for the invitation. I'm plugged in. Perfect. All right. So I have my own computer here because I have we're going to try a live demo. It has worked everywhere I've tried it, which means it'll probably fail today, right? So I'm a botanist. I did all three of my degrees here at UW-Madison. I'm now an adjunct prof in the botany department, so I am as academically inbred as it's possible to be. I like to make the joke that if you read my CV, you can hear banjos playing in the background. I'm an employee of the Forest Service. I work at the Center for Wood Anatomy Research at the Forest Products Lab, and so I'm actually paid by the Forest Service, and what I do with the university affiliations is as a volunteer. I'm living the dream because I am a professional nerd. I mean, I get paid to just be a nerd, right? It's what you're all trying to do. And so I kind of stuck the landing. So I want to talk about that. I'm going to kind of take a different take on things because a lot of my program is about combating illegal logging internationally. And so we try to come up with tech that's cheap and DIY and hopefully useful. And so technology, for those of you that were at the Broderson talk yesterday, I have proof with a group of colleagues from last night that I did not steal this slide from him. This is what I want to open with, is that I can't stand still, I'm sorry, I'm a pacing guy, so if the technology that you need or you want or the method that you need or want doesn't exist, invent it, right? Figure it out, develop it, modify it, figure it out. And if you can't do it, find somebody who you can partner with who can. Don't leave something on the table just because you don't know how to do it now, right? If we're going to talk about technology, also go for best fit technology, not necessarily the sexiest or the newest or the fanciest, but what works and solves your problem, right? Most of what I do, I can do with a hand lens or a simple microscope, and that's the tool that's most appropriate for what we're doing. So again, Forest Products Lab, a big ugly building on the west end of campus. I've actually worked there since I was a freshman in college, so I was grown from seed by the Forest Service to do this job. It's the only thing I know how to do. And in terms of like a little historical note, my academic lineage in the Forest Service, my great-grandfather was Arthur Kaler, who was the expert witness for the first use of botanical forensics in the country and the expert witness in the trial of Bruno Hauptmann for the Lindbergh kidnapping, which put forensic botany on the map. So I actually have his old microscope. I don't use it. I have a much nicer, better one. But I've been doing this for almost 30 years now. So I said 348-year-old tech in my title, and that's true because the first simple microscopes were basically hand lenses. And the amount of variation, the amount of the world that you can see with just 10x, 14x magnification is amazing. So this is a woodcut from a letter that Wiedenhoeft wrote to the Royal Academy of Dutch Science in 1676, seeing a lot of the same kinds of features that we still use today, a lot of very similar magnification to some of the technology I'm going to show you. And I'm coming at this from a standpoint of wood identification. And historically, we've done wood identification based on wood anatomical, plant anatomical, structural features. So when I look at a piece of wood with my hand lens, I'll see something like this, and I can see some amazing patterns. I'll show you some as well. More recent technology will focus on molecular variability, right, with various forms of spectroscopy, spectrometry, DNA analysis. And so we have these two sets of variability in wood. We can look at structural, we can look at molecular, we can look at both. But if we want to do things that are cheap, if we want to do things that are fast, we probably focus on the structural. We can have interesting arguments about that back and forth. And if you look at this image here and you think, wow, that's really beautiful and it's amazing, you're right. And that's just one image of one piece of one species. It's one tiny drop of what the variability of wood looks like structurally. So regardless of which form of variability we're interested in, we have this critical need for technologies to enable us to do wood identification. Ideally, enable us to do it easily, cheaply, quickly, and in the field, if possible. Now, a lot of people, I'm not going to call for a show of hands, might not have even heard of wood identification, right? They wouldn't think that there's a forensic botanist or a forensic wood anatomist. So I'm not being dramatic, though, when I say that there's a critical need for identification expertise. We did a study that we published in 2019, and we showed that almost 2/3 of normal wood products that people would go buy in a store are fraudulent or misrepresented with regard to what kind of wood they actually are, whether or not they're solid wood. Now, I've known this my entire professional life because those of you who've seen me before, I don't go anywhere without my hand lens. So if I'm at a store, and I rebuilt an old farmhouse back in the day, and so if I'm waiting to have carpet cut, I will identify every wood specimen in your demo flooring section and figure out, oh, that's illegally logged from Southeast Asia. That's an endangered species. So I've known this my whole career. We did a study to actually show that it's true. And so to do the study, we worked with some NGOs who went out, spent about $14,000 on wood products, cut them into little tiny pieces with just a number on them, and sent them to the lab. Then we identified them, botanically what they were. Then after that, the claim on the product was revealed to us, and we evaluated whether or not that was consistent. So that's part of where that information comes from. And so the graphics for the colleagues from the World Resources Institute put this together. 55% incorrect botanical, 21% product type. So that would be like, it says it's solid wood, but it's actually veneer glued onto MDF or particle board or something like that. So we knew that this was a problem. We were able to quantify how much it is a problem. Then another part of this study was we sent out surveys to all the other laboratories that purport to be able to do wood identification at a competent level. And we did a proficiency test. And grading extremely liberally, accuracies ranged, total score on the test was as low as 6%. And that was an academic institution teaching wood identification to students, and 92%, which was actually a independent contractor person. And that's grading so liberally that we sent them a bag of 55 specimens, and we said, okay, if you know you don't know what it is, we won't even count that in the grade. So you just go, well, no, not, okay, fine, we won't count that against you, don't lose any points for that, because you have the sense at least to say, I don't know. Okay, so I think it's fair to say that the technical term is pathetic for these results, and so we don't have the capacity to do anything close to the kind of work we need to do as an importing country who moves all this stuff around, and so we need technology for legal compliance and law enforcement, and that's something that I want to point out because we have some industry folks here, which is nice. Because I think sometimes in an academic setting we lose sight of that. I come at a lot of this technology to combat illegal logging. And one of the ways to do that is to catch the bad guys. But another way to do that is to make sure that responsible industry, responsible players have what they need to have verifiable supply chains. And they can conduct business with scientific backing at the speed of actual trade. So we have to do both sides. And because I'm federal government, I can be on both sides of that at the same time, which is kind of nice. So I want to tell you a little story here. In 1999, when I was younger than a whole lot of you out here, just a kid, I was asked to develop a field manual for the CITES-controlled timbers, right? The Convention on the International Trade in Endangered Species. And back then, there were only 20-some species, and my mentor, Regis Miller, and I, we were asked to do this, and we worked for a couple years. It took a while. We published in English, French, and Spanish, and it was later translated into Polish, into Chinese. It was distributed to 160 countries around the world. I was so excited. I was so proud. It was the first time I had done anything like this, and I thought we were really making a difference, right? We put this weird knowledge that only we have, and we got it out there, and it's in 3 languages, and if you just read the book and actually do what it says, you will learn how to identify these woods. And it was crickets. No one cared. Nobody said good job. Nobody used the book. It took about another 5 years, and then countries started calling up and saying, look, we know you wrote the book. We know all the information's in the book, but we're not going to read your book. So how about you come down here and teach a course so that we don't have to read the book? And I'm a sucker, so I did it. And so, and it's the ill-fated trainings that I want to talk about. So in 2007, I went to Nicaragua. I trained a group of 30 people for a week. I had tons of fun. I love Spanish. I love Nicaragua, it turns out. It was my first time there. I love teaching this stuff. And there had just been an election, and by the time I went to Honduras later, I went to Singapore. Within a couple months, everyone that I trained in Honduras but one guy was fired because he was the wrong political party. A couple months later, Honduras had their coup, and a bunch of the people that I trained were killed. And then Singapore went great. Then Nicaragua, with no sense of irony, called me up and said, hey, we fired everybody. Would you come back and do it again, please? And I did. But then I came back and I'm like, okay, I can't do this. I can't train human beings fast enough to make a difference, right? Because you get a group of 30 people together to learn something weird like wood identification, and 1 or 2 of them, for 1 unit of effort, are going to make 3 units of progress. Most of them are going to make 1 unit of progress with 1 unit of effort, and some of them are hopeless. And they're going to make virtually no progress with 3 units of effort. Not because they're not smart, not because they're not trying, just because some people are just no good at something. I can't dribble a basketball to save my life, right? I've tried, didn't work. So I stopped, right? And so that's another little soapbox aside, which is you're in graduate school, figure out what you're really good at and you enjoy and how to make that work. If you look and say, I'm really good at this side of stuff, then find a way to emphasize that. Take advantage of the things that you're better at. You still have to work on the stuff that you're not good at. You still have to learn all that. But don't force yourself down that path because you're going to be miserable. That's an aside. Okay, so back. We need a technological solution. So our vision then for my team is we want to develop, at least with regard to the forensic stuff, we want to develop tools and technologies that enable compliance with laws and permit and also empower law enforcement, right? We want to make it so easy to comply with the law that only bad guys break the law, right? Because if it's impossible to follow the law, that's a bad law, right? I mean, I don't want to get down that road of bad laws, but you can't expect, let's say, industrial compliance if there are no tools in existence that they can use to make those decisions. So we want to be on both sides of that. And that was really the genesis of the Xylo platform, is can we teach a computer to identify wood? Because human beings sometimes are no good. Practice for a week, but then never practice again. Get hit by a bus, retire, get promoted, right? These things happen. So human expertise, it can be labile, it can be really fragile. So if we can teach a computer to do it well, it'll do it the same way every time. And can we get it to do it in the wood? In the wood. That's where my brain is. In the field, right? to be able to support supply chain management and law enforcement. So what the Xylo platform is it's an automated wood identification system. And we ended up with this kind of lightsaber looking thing on the lower left, and that's the original Xylotron. And then the xylophone on the right. And that's basically a better solution to the real world problems, but we'll get to that in a second. So the initial idea was, can we just get a smartphone back in like 2011, just take a picture with that and teach it to identify wood? And the answer was no. We're not at the right magnification. We can't control the lighting. Back in 2011, cell phones were not strong enough to do this. So we pivoted to this idea that we need to create our own peripheral, our own imaging device to control all of those inputs. And so the first version, we're trying to do all this open source DIY as cheap as possible. So we started with the pipe bomb version, which was PVC tubes and fittings and all that. TSA, not a fan. Hard to travel with this one. I've got a pipe packed with a bunch of wires, electronics, and metal things. Let me on the plane. What's the problem, right? But it also mechanically it didn't work. It was too fragile. It fell out of calibration. So then we tried the Maglite version, the big police flashlights, because you can just buy those and rip the guts out of them. They're aluminum. They're strong. That's an even worse pipe bomb. Not cool. And also didn't work out great. Everything fell out of focus, and it was too easy for people to futz with the innards, and we don't want that. We don't want you in there messing with this originally. Now, with what I'll get to in a second, you have to build your own, right? So if you're a Star Wars fan, it's like the last step to be a Jedi. You have to build your own Xylotron. We had an aluminum cage version where all the components were in this really weird cage here, and we had to focus it with all these screws, and it doesn't matter. It was dreadful. So that we didn't like that. We came up finally with this version, and we published this open source. We distributed these to research partners around the world, to other xylaria, other wood collections. And it takes really nice pictures, like the one I showed you before. It was a mechanically poor design, and so what happened was I was working with the Forest Stewardship Council on their supply chain stuff, doing forensic analysis of FSC certified stuff, figuring out when things are not what they're claimed to be. And the EU needed charcoal identification capacity. And so the question was, can we take the Xylotron that's made to image wood and use it to get the same features out of solid charcoal? And it turns out that what we needed to do was move the lighting array, instead of right next to the wood, to be as far from the wood, which it's a long conversation, but it makes really good sense once you figure out that that's what you needed to do. And so I designed a new Xylotron with the lighting array way far away. So we've got $1,300 worth of lenses and cameras, and now the lighting is over here and we can't use it for wood. Or we've got the same cost configured for wood and we can't use it for charcoal. That's a lot of money to spend on a thing that, other than this one factor, could theoretically do both. And as an aside, UW and FPL share a patent for this that was just granted for using this kind of technology for charcoal. And so that gave us the need for the Xylotron 2.0. So basically, this component right here is the UV lighting array, and it's now movable along these metal rails to be in the charcoal position, which is what's depicted here, or if it's mated down with the front cap, in the wood position. And so now with the same device, we can do both substrates and not waste all that money. And so the original designs for the printed circuit boards I actually did on paper, and then I was very lucky to have Dr. Blaise Thompson at UW Chemistry, who's an actual grown-up and professional with this stuff, turn that into real PCBs. That was excellent, because I'm just a plant anatomist, so I don't only have so much business working on this stuff. But we published it with a step-by-step, color-illustrated, like 40-page DIY assembly manual. How do you actually put this together? Which wire do you solder to which one, so that, in principle, anybody in the world with the components could do it? The Xylotron requires a laptop, which means it's a lot of cables and it's a lot of cost. As an aside, and then this was a stainless steel cage. I came up with a 3D printed cage, which I actually happen to prefer. And what you can do with this then is you can do macroscopic imaging of basically any substrate. So we've got wood, charcoal, developing gills of, actually, what's up there is, like the button mushroom. That's actually a portobello in the image, but that's okay. Feathers, textiles, what have you. Anything with interesting macroscopic variation. And then we have open source software for collecting reference images, for actually doing the identifications, and for just using this very expensive thing as a glorified hand lens, which seems silly, but students actually really like it way more than picking up a hand lens and using their eyeballs, which is frustrating for me, but is phenomenologically true, so what are you going to do about it?
Normally, I'd bring the Xylotron in and show you guys in person, but right now we've got it configured here for automatic scanning of large specimens to be able to capture high-resolution images and stitch together, which I'll show you in a minute. So instead of showing you the Xylotron, I'll show you a quick video of how I used the Xylotron when I testified in a murder trial, and then I'll show you the kind of the second generation of the Xylotron, which is a smartphone-based one, which is the Xylophone instead.
So this is a home, if you can't tell, this is a home-built device, but it uses a $13 motor from Amazon and a $20 controller. So it's, it's low budget, it's low rent, it's held together with a C-clamp over here. But I can actually put that whole thing on a surface. I could come up and scan the floor if I wanted to. So it's versatile. It has things that we can do with it. One of the things is I've got this specimen of bristlecone pine, right? So from here to here on this specimen is about 900 growth rings. And using this, I imaged that this is the stitched together image then from one end to the other of that bristlecone pine. And if we put this up full screen and then we go full resolution, we start at one end and you see how tiny some of those growth rings are. And we go all the way to the other end. And I did the whole scan in something like, I don't know, like 1.5 minutes or something. So it's pretty, it's low rent in the sense that it's a low budget, kind of simple thing to do, but it works pretty nicely. Now I'm gonna give you the very quick rundown on the murder case. So right before COVID quarantine, I testified in the murder trial, and murder cases don't make sense. So I'm gonna give you the super fast version. In 2010, this guy decided to kill his 16-year-old son's girlfriend's mom. Doesn't matter why, okay. He decided he was going to dispose of her body on a burn pile, a brush pile out in the back 40 on his rural property. And his son saw him do it, and he made his son help in the body disposal. 8 years later, this woman just goes missing. 8 years later, the same murderer dad guy decides it's time to kill his own wife. And the son is not cool with that because he likes his mom. And so he actually gets immunity and testifies that, oh yeah, we took Bonnie Woodward out back to a brush pile and burned her down to nothing, scraped what was left into the bucket of our tractor, took that over to the river, dumped it in the river. So they did an outdoor body recovery, which is like a high-speed archaeological dig. They found one piece of charred bone that they didn't even know is mammal bone, and that's it. No forensic evidence, no physical evidence. But the investigator says, well, look at this tree. I wonder if that tree can tell us something. And so that's where the wood anatomy is that the tree was actually a witness to this crime. And they cut down the tree for me and sent it to me. Now, Bonnie Woodward went missing in June of 2010. They cut down the tree for me in June of 2018. And so what I did is, here, we should get another video here because. So the alleged fire happened in June of 2010. And this tree was cut down. It was alive at the time it was felled in June of 2018. So if we start at the outside, this is the outer bark, and we move inward to the inner bark, and then this is the partial growth ring that was being formed when the tree was cut down in 2018. So this is 2017, 2016, 2015, 2014, 2013, 2012, 2011, and then 2010. Now we move laterally in the 2010 growth ring toward the site of the wound to see whether or not the wound lines up with having occurred during the 2010 growing season. And here we get to the margin of the wound. And you can see quite clearly that part of the growth ring of 2010 was formed. Then we have this wound. If we continue over here, you can see some of the growth ring in the uninjured part was still formed afterwards, so it's consistent with the middle of the growth ring wound. And then this is wound healing tissue up here. And this process that I just did, showing you this with the xylitron, which I can tip the camera here, is exactly what I did in the courtroom. And actually, they let me keep the piece. So this was People's Exhibit 130. That was part of this case, and it came back to the laboratory with me. So that's the xylitron. That's a weird use for it. We also use it to do automatic field identification of wood. But one of the things we learned is carrying a laptop around with all these cables is really burdensome. It's difficult. It's not very agile. And you can't just take it out into the tropics where it rains. So not super practical. Great for collecting research images, great for teaching, great for things like this, not super practical in the field. So that's where the idea of the xylophone came for. We wanted something smaller, cheaper, scalable. And since smartphones are almost ubiquitous around the world, the question was, can I develop a peripheral device that controls that illumination, that gets the magnification that we need, and that we can adapt to, let's put it in quotes, at least any smartphone. And so this is what I did. So I came home from that murder trial, and like 2 days later, we went into lockdown. And I'm a plant anatomist, so this was like soul-crushing iterations of spectacular failure over and over and over again, because I had to learn about the electronics and learn about 3D design and 3D printing and all that. And ultimately, though, I used we popped the lens out of one of these Belomo 10 Hastings triplets and used that as the magnifier. And this was published open source, and again with an assembly manual. And I don't see Cal or Aishwarya here, but they just helped update it. They just built their own and updated the assembly manual with some new tips and tricks. And so this is about 10 times cheaper than a xylitron. Again, we can image anything that has interesting macroscopic variability, whether it's wood or mushrooms or what have you. And so I'd like to show you guys. Where it's working. Ha. All right. So that's my shirt, right? And this is our bristlecone pine that we were just looking at a minute ago. Same kind of look. And just because wood is spectacular and you probably have never seen it before, this is a Brazilian South American wood, Rupala proteaceae. Here's totally different pattern. So this is what I get to see when I look at wood under the microscope. And here's a real banger. It's spectacular. The world is so beautiful just at this magnification. But what I'm really here to show you is the Philippine Wood ID app. So I've got specimens of wood. This is developed by Joseph Dio. And actually, I love this stuff, and so I get excited. I think the slide for this is coming up in a minute, but we're here, so we're doing it. So all you do is put it on the specimen, get those little lines called rays running vertical, hit identify sample, and it spits back. So that's the image we just took, and it says it's apitong, which is Dipterocarpus, which is what that specimen was, and it gives us the ranked list of what it thinks is next. So we close, and this is happening on the device. It's not the cloud, it's not anything else, because if you want to use this in the developing world, for example, you don't necessarily count on having good internet access, especially at a control point on the highway or something. Specimen of teak, it gets teak, and then for whatever reason, the Filipinos import ash from us, which is not going to last long with emerald ash borer. But so here's a specimen of ash. And it comes back and it thinks it's teak. Now so this is an interesting error, because if you look at these two images here and here, They are similar in some ways. They share some wood anatomical characteristics. One of the things that we're having fun with our stuff is that we're getting sensible errors when we get errors for the most part. Sometimes, though, AI gives you spectacularly unreasonable errors. And that's, I mean, I'm not gonna, like, if you know about like ImageNet stuff, right? For one, machine learning people will say, well, if it's in the top 3 predictions, then I did it right. That doesn't work in real life. If you're at a control point and you have to say yes or no on this shipment of wood because it's your one chance to stop it, if the machine gives you 3 answers and one of those is what it is, you don't have a rational way of knowing that unless we use like a human in the loop system, right? So in this case, I can see the image. What I'm going to ask Joseph to do next is, here's my image. Give me a reference image of what it thinks it is. So as a human being, I can look at it because, you know, some of the ImageNet models will be like, my top prediction is it's a duck. My second prediction is it's a cat. My third prediction is it's a fire truck, and it's actually a cat. And you go, whoa. I mean, those are disparate, right? Because if it were a cat and you got a dog and a squirrel, at least you're 4-legged mammal, right? So sometimes we get some pretty crazy failures. So having a human being a part of how we make these decisions is valuable. So I just did the demo there with the Philippine Wood ID app. And the other thing that's kind of fun with this is you can use it for lots of other things. My colleague, Joanna Ostapkovich at Oxford, takes this to museums to look at wooden cultural artifacts that can't be sampled, and she can send me wood anatomical pictures like this. I can see some features here that help us make some inferences. It's great for teaching. I used it a lot this week in dendrology to show students things, so we're looking at the same things at the same time. And so anything that has interesting macroscopic variability, it can image. One last piece of tech, and then I'm going to close out, is a colleague of mine and I just redid my 2011 Central American manual. We added the whole kind of biogeographically relevant region. And so now it's this monster tome. This is a field manual, okay? Right, that's very practical. So we have a graduate student, Kayat Spessert down at Mississippi State, who has turned this into an app. But, okay, it's letting me do it. Cool. So, and that's right here. And the thing that's cool about the app is that you can go to the text and see the chapters in the book just like it's laid out, and it'll keep track of where you are. You can take skills quizzes if you haven't learned the wood anatomical features yet. Which of these woods is showing diffuse porosity? Questions like that. And then you can actually go into the key, and if you don't know which one you want on the key, you can tap on it. Oh, I gotta choose that first. So I make that selection. It takes you immediately to the next selection. Then let's say I say this, and it takes us to bamboo right away. And then it reminds you, compare it to a real specimen, or at least go, and if we want to see the pictures bigger, we can click on them. Way better than an actual book. And then it just fits on your smartphone, and you've got it everywhere. So simple things like this. Not, obviously, an app is not that fancy anymore, but it is a technological improvement over carrying this thing around in the field. Nobody's going to carry this anywhere. We only printed 200 copies because it looks cool. Other than that, there's no point. So I think that's just to reiterate the message. I kind of want to end where we started, which is if you don't have it, figure it out, invent it. If it already exists and it works for what you need, if it's not broken, don't fix it, right? Use it, adapt it, figure out new ways to deal with the data, all of that. But don't necessarily chase the new shiny thing unless it helps you solve a problem that you can't otherwise solve. So with that, I'd just like to thank everybody for the opportunity for this, the organizers and all the sponsors and supporters, all of my cooperators, colleagues, friends, family who have made all this possible. And also, I want to thank all my detractors. And because I mean it, I'm not just trying to be, trying to be funny, but it's not only that. At a certain point in your career, if you don't have haters, you're probably not sciencing properly. So, but there's a lot you can learn from them. So it's really valuable to have that in your life. So with that, thank you very much.
Thank you, Dr. Wiedenhoeft. I've seen Alex speak before, but each time it's just as engaging. It's just incredible work. So we now invite questions from the floor or from Slido, which you can access using the QR code on the table. And you can also just raise your hand and we'll bring a microphone to you. Great, I see some hands.
Thank you. That was a really awesome talk. So I have a couple questions.
I'm
First,
sorry, where are you?
I'm right here.
I just want to
Yep.
pretend I'm
So,
making eye contact. Yeah.
First, my husband's an arborist, so I know what I'm getting him for Christmas. How do I get access to like the plans for the xylophone so that we can build one at home?
Yeah, it was published in the IAWA journal, the International Association of Wood Anatomy Journal. It's open access. And all the files, the 3D printing files, the PCB files, everything's in the supplemental information, which I think is actually just posted on our GitHub link, not through the journal itself, but the link to that is in the paper.
Okay, awesome. And then second, does this xylophone, does it work better on like dried wood versus like more recently harvested samples, or like do you have to have the wood cut in a certain way for it to be most effective, or can you talk about some of the more practical applications like?
Yeah, no, that's a great question. So our stuff has been developed on xylarium specimens, so scientific reference specimens that are dry. They're at like 6 to 8% moisture content. Mostly it's getting used on things that are green in the real world. And wood changes size and shape as it shrinks and swells with changes in moisture content. So we've actually looked at using image augmentation because we have known shrinkage values for lots of woods. So we can actually use the known shrinkage value to digitally manipulate, right, the size of features. So that's an approach that we were planning on taking. But so far with real world testing, we haven't seen. We talk about okay. When you tell a machine to go sit in the corner and come back with an answer and it's going to tell you how smart it is, that's like having your undergrads grade their own exams. It will come back to you and tell you, I'm amazing at this. And what you have to do, or at least what we've found, is you have to challenge it with a completely unique data set to see if it's making correct predictions. Because no matter how hard you try, you end up with some kind of overfitting or some kind of less generalizable model. So it's really good at the specimens it's trained on. When it sees new specimens, it's less good. So we talk about the difference between what it tells you and what we see when we actually verify is the deployment gap, right? That gap in accuracy between the real world and kind of science, right? And we haven't seen the need to worry about that much so far with the way we've been training our models. They seem pretty robust to that. But yeah, it's something that we're prepared to handle if we have to. Yeah.
That's awesome.
And do you have to prepare a specimen a certain way? You do. And actually, maybe it'd be more fun to just show you. Because if we go back to just the camera version, so I want something with a raggedy here. So this is no, that's not bad enough. The problem is I have really good saws in my lab, so all the saw cuts look really good. Oh, here we go. This is pretty dreadful. That's the wood anatomy of a saw cut. Okay? So I'm going to I brought a knife just in case we're going to do this. Take that same area. And now we have anatomical features. So you have to cut the transverse surface, and you can also polish it in the field with a rechargeable sander or something. But if you can do the cut and end up with the same number of digits as you started the day, that's the faster way to do it.
Fantastic, thank you. We have no more time for questions right now, but there will be a Q&A at the end of the day with all of the speakers. So thank you everyone for your questions, and let's give another round of thanks to Dr. Wiedenhoeft. Okay, great. Thank you. Awesome. So it is time to transition now to our lightning talk stage. And so if our lightning talk presenters will come up and now, and you can come up and you can arrange yourself in the order that I'll say on the left side of the stage so that when the time comes, you can just file in. And so, well, once you get here, I'll tell you which order to be in. So we have 6 brave researchers that will tell the story of their research in 3 minutes or less. This is a competition, and the winning presentation will be selected by you, the audience. A QR code for voting will be displayed on the screen during the break and directly after these talks. The audience-selected winner will receive the grand prize of $125. The second place winner will receive a cash prize of $75, and our third place winner will win a cash prize of $50. Please save your applause towards the end when all presenters have finished. So first up, we will have Fernando de la Torre from the Department of Plant and Agroecosystem Sciences in the Plant Breeding and Plant Genetics Training Program. So without further ado, please join me in welcoming our first presenter.
Thank you, everybody. Yes, so consider the following 3 fruits, the tangelo, the plua, and the boysenberry. Now, these are hybrids selected to combine the unique fruit quality traits of their parent plants to create an exciting new fruit. For example, the tangelo combines the size of the pomelo, which is the largest citrus fruit, one of the largest citrus fruits known, with the juiciness and the color of the tangerine. The plua takes the skin and the internal color of the plum and combines it with the texture of the apricot. And the boysenberry takes the color of the raspberry and combines it with the size of the blackberry. Now, with this in mind, I want to introduce you to my objective of combining blueberry and cranberry for their complementary fruit traits. So, top left, you have a grocery display of blueberry, blackberry, and raspberry. And so what you might notice is that there isn't necessarily a red berry of comparable size on the market other than the raspberry, which is sweet but has poor shelf life and low firmness. And this is very similar to the case of blueberry. Sweet but poor shelf life and low firmness. Now, consider cranberry, which is related genetically to blueberry. It shows the opposite. It shows low sweetness but high shelf life and high firmness. So therein lies the opportunity. In our USDA ARS lab, we've been able to generate cran-blue hybrids that can combine the best of blueberry and the best of cranberry. So you can see with these pictures down here that some of the plants, some of the hybrids produce fruit that is more like cranberry, and some of them produce fruit that is more like blueberry. So our most exciting results are for firmness and soluble solids. So on those curves up there, you can see that cranberry firmness is high, blueberry firmness is low, and hybrid firmness is intermediate. Furthermore, we see something similar with soluble solids. So this graph on bottom right, we see that cranberry alone across many different cultivars can only contribute so much to soluble solids, and it's only through the crossing with blueberry that you can finally break through and produce red berries closer to the sweetness of blueberry. So in summation, can this innovation encourage the consumption of smaller fruits? Possibly, but will this new berry pique the curiosity and expand the palate of consumers? I think so. So stay tuned.
Wonderful, thank you. And now we have Julian Cooper, who is one of the recipients of our new travel grant program from the University of Minnesota in the graduate program in Plant Applied Science.
Hi, everybody. Thank you for having me. My name's Julian Cooper. I'm finishing up my PhD with Dr. Corey Hirsch. And when I was first starting out in graduate school, I was told your results will only ever be as good as your data. But when you're phenotyping Fusarium head blight in wheat, that's kind of tricky. It's got very nondescript symptoms, a narrow rating interval, and rating itself is time and labor intensive and can be subjective based on the rating method used. To combat that, we spent the last 4 years developing an RGB-based deep learning image analysis pipeline for phenotyping this disease. It started in 2021, where we partnered with Mineral, a Google subsidiary, and used their phenotyping rover to image wheat throughout growth and development. We then took those images and developed our deep learning-based pipeline that had 4 models. To 1, detect wheat spikes in complex field conditions. 2, assess the image quality of those spikes. And then 3, quantify disease on a per spike basis. But remember, this is a tricky disease to rate in the field. We wanted to be sure we were really measuring what we wanted to. So in 2022, we went through a very robust validation method, both with raters in the field and raters annotating individual images to mimic pipeline outputs. We found that our pipeline was accurate at the individual spike and plot scale and was robust across locations, as you can see on top, and disease progression throughout time. But there's an issue here. We've talked about it today. This large phenotyping rover is not very accessible, and I believe the company is now also bankrupt. So this can't be used for other research groups. So what we wanted to do is translate this to use mobile images. We took the same image analysis pipeline developed in 2021, and we saw a 0.96 correlation between disease inferences from the rover and from mobile images. And interestingly, we actually were able to detect many more spikes per image using mobile phones. One problem, though, is this whole thing has been done and validated so far on paired row plots, where we're looking at the same genotype. That's not really how researchers and breeders will grow. So in 2024, we're really working on improving this pipeline to work on single row plots, so we can image multiple genotypes at once. Unfortunately, as people who maybe use deep learning models know, the size of your image, the orientation of your image has a big impact on the quality of your predictions. So we're training a new mobile image specific gradability model just to really make sure we're measuring the spikes we want to, make sure image quality is still good. Very preliminary results, but we do see pretty decent correlations between our mobile image analysis pipeline on single row plots with raters in the field, and importantly, there's a high correlation between mobile users imaging the same plot, which allows us to scale up, and we hope to introduce this pipeline to new research groups starting this summer, 2025. Thank you very much.
Thank you, and thank you for coming all the way from Minnesota. Next up, we have my own lab mate, Zoe Ryan from the Department of Botany.
Thanks, guys. Let's see if does the pointer thing work? Oh, awesome. Okay, great. Okay, I'm here today to talk to you about my master's thesis on niche modeling and distribution modeling of bryophytes. So this gorgeous sparkly species that you see right here is Entodon seductrix. It's a moss that's common throughout Wisconsin, Illinois, and the rest of eastern North America. This moss is just one of the 20,000 species of bryophytes, which makes them the second largest plant group and have species present in every environment all over the world. So not only are they great contributors to plant diversity, but can be very useful bioindicators, especially with the oncoming threat of climate change. However, it can be difficult to understand the full range of a bryophyte species because they're very sensitive to environmental changes and relatively small. So my project attempts to look at some common Illinois bryophyte species using species distribution modeling methods to see if we can determine the current range of these species, what it might look like with the threat of climate change, and then what environmental drivers are really creating these distributions. So you can see here in the image on the right, this is our model of Entodon seductrix. This was created through MaxEnt by putting in just occurrence data, climate data, and land use data, so very simply based. This is a heat map showing that distribution likelihood with those green areas representing high likelihood for our species. So we see the southern Illinois forests, along the Illinois River, and the Chicagoland area as well. And we can see that this is well reflected in the real world as well, so it's always good to see models turn out the way we want them to. Over here we have a jackknife plot, and this is what is telling us what environmental drivers are determining our distribution, what's really important to our model. So these dark blue bars up here are our most important ones here, and that is land cover and canopy cover, tree canopy cover. So we're really seeing this drive of land use for determining our bryophyte species rather than climate in Entodon seductrix and in the other modeled species. Then for this climate change aspect, we can compare our current base model to a future model. So this was created the same way, but with future projections of climate data. So this is an estimate of about 2050 if we're on our current carbon emissions path. And just looking at some of the different colors and areas, we can see in these red circles that we're starting to lose some of our green, we're starting to lose some of our distribution, which might be a little sad for our Entodon seductrix, but that could mean it could be a good bio-indicator, right? A good climate change indicator. So tracking the growth and distribution changes of this species of a proxy of how climate is altering our environment. If you're interested in models like this, I have a poster right out there that can show you how to do it as a beginner completely for free. Thanks, guys.
Wonderful. Thank you, Zoe. And so next up, we have the other recipient of our new travel grant program, Ashley Barstow from North Dakota State University in the Department of Plant Sciences.
Every day, researchers generate mountains of genomic data. But with all that information comes a critical question. How can we sift through it to find valuable genetic markers? And just as importantly, how do we filter out the noise? Accurate variant calling is vital in understanding the genetics of complex traits. Traditional filtering approaches, known as hard filters, set strict thresholds to weed out low-quality data. While they reduce errors, they can also inadvertently discard genomic variation linked with the phenotype you're studying. Our previous research failed to identify an interval containing a strong candidate gene for nectar production in sunflower. We hypothesized that rigid hard filters were excluding crucial markers. That brings the question, could there be a better way to identify reliable markers without losing meaningful genetic data? Instead of arbitrary hard filters, I used a new approach, a biologically relevant filter that focuses on how markers are expected to segregate according to Mendelian genetics. This approach involves applying a chi-square filter to retain only the variants that fit that expected genetic ratio. To validate this approach, we used that data set and we tried remapping nectar production-related genes in sunflower. We found that not only were we able to generate a larger number of valid markers with better genome coverage, we were also able to identify more candidate regions with nectar-related genes, as seen in blue. In a world overflowing with genomic data, a key challenge is knowing what information to keep and what to throw out. Traditional filtering approaches can discard valuable genetic markers, limiting our understanding of complex traits. My research shows this by replacing those rigid hard filters with a biologically relevant filter that aligns with Mendelian genetics. We were able to identify strong candidate genes for nectar production in sunflower. This method not only retains crucial markers, but it also opens up new doors for plant breeding. Thank you.
Thank you, Ashley, and thank you for coming all the way from North Dakota. So next up, we have Gopika Gopinathan from the Department of Plant and Agroecosystem Sciences in the Plant Breeding and Plant Genetics Training Program.
I'm sorry for the small title there. I just thought like I need only 1 to 2 slides, so I just fit it in one. So yeah, my topic is speed breeding in barley and protocol for covered spot disease screening. How does this move? Okay, so barley is the 4th most important cereal grain in the world, and very importantly, it is used in beer production, as we are all interested, and in the brewing industry. As breeding every crop, barley varieties are also released initially with a cross made, and And then the generation advancements follows, and then multiple location and multiple year testing is required for a release of a cultivar. And this whole process might take 9 to 11 years in conventional method. So we thought of having a method which can reduce the time for the generation advancement, which is called speed breeding. We used 22 hours of light and slightly higher temperature at night for this. And what we saw, so we had two growth systems of study. And what we saw was like we planted the seeds and we recorded the growth stages of barley in like the different growth stages. And at flowering, we had a, like that is a means we had. And in the two growth systems, we saw that out of the 11 genotypes of barley we had, 9 genotypes had reached flowering earlier in speed breeding system. So we knew that speed breeding is working. And we also wanted to evaluate the effect of early harvest to further decrease the time of the breeding cycle. And we harvested in an experimental unit at 3 different days after flowering. And interestingly, we saw that out of the genotypes we have, at 21 days after flowering and 28 days after flowering, when we harvested, we had a high germination percentage. And we also had like at least 5 viable seeds, which is enough for advancing the next generation. As 21 days is less, we concluded that speed breeding with 21 days after flowering harvest could give us 4.5 generations per year, which is huge when compared to the normal system where only we could get 3 generations per year. And I want to dive into my next topic, which is related to organic systems, because there is a huge demand in organic agriculture these days because of high demand in organic food products. And diseases can be a very vital problem in organic agriculture because of lack of use of pesticides and fungicides. One such disease in barley is covered smut. It decreases quality of seed over storage. And we wanted to develop a protocol for this because this disease has not been studied for a very long time now. But increase of organic agriculture, increase of the disease. And we have to develop disease-resistant varieties for the same. So we wanted to have a protocol, and we tested 3 different inoculum concentrations in speed breeding in 20 genotypes, and we saw that several genotypes did not show any disease, and in the ones which there was disease, there was a susceptible check which showed high disease, and then there was another genotype which was showing a higher infection. But in general, you can see that the infection percentage the maximum was 30%. There was not a lot of infection. And we thought why this is happening. There is some disease escape mechanism. So we did a power analysis to understand how many plants we need to see at least one infection. And as we are still in the protocol development stage, we saw that we need at least 74 plants per treatment, and all the infections we have here is just like luck. And we only had like 24 plants. So, and also like the fungus is growing slower than the plant itself. So we would go for the normal growth conditions rather than speed conditions. And right now we are doing this in the greenhouse. So yeah, by this we are expecting to get a protocol to screen the disease effectively. Thank you.
Thank you so much, Gopika. So next up, we have Francisco Campos Arguedas from the Department of Plant and Agroecosystem Sciences in the Horticulture Training Program.
I don't know how to start this. All right, so good morning, everyone. I wanted to welcome you to a warmer future today by exploring cold hardness and dormancy responses in boreal forest species. So I want to start it off by explaining what winter physiology is. So I work with buds. So buds are set during the during the summer, sorry, and in order to grow during the next spring, they need to survive winter and fall. So they go to a process called dormancy. At the same time, they have to achieve cold hardness. So they acclimate during this period. Once temperatures start increasing again during the spring, they start deacclimating and then they end up with bud break. So my project is located in northern Minnesota. We have there different temperature treatments and we control for CO2. So we have big enclosures where we control it from 0 to 0.25, 4.5, 6.75, and 9, kind of like mimicking devastating climate change scenarios. And then I have two main species, American larch and black spruce, one a deciduous conifer and the other an evergreen conifer, and these will become important in a little bit. So the way we're gonna look at the data here is gonna be on the x-axis. You're gonna see like the month of the year, and on the y-axis you're gonna see temperature. The ribbon that's there, you're gonna see minimum max daily temperature for each of the treatments, and then you're gonna see a line that looks like that, like a smiley face pattern. That's gonna be the cold hardness that we measure. So right here, I'm just showing the temperature patterns for the control plants, which are no warming at all, and the ones that have received the warmest treatment, which is a plus 9 degrees increasement in temperature. So this is how cold hardness look for the control plants. I'm gonna explain a little bit in a second, and then this is how the warmer ones look like. I wanna focus on three important moments: fall, winter, and spring. And then I just wanted to highlight here that last season, during 2024, in January, there was a really warm spell that if you see the graph there, just comparing the two cold hardness measurements in this moment, it triggered the loss of cold hardness of the species that were exposed to warmer temperatures, but that didn't happen to the control ones. So here I just show a graph where it has like the whole range of temperature treatments that I have and the responses that they have, just to kind of see that the responses vary to the degree of warming that we're applying. And then here, I just wanted to highlight that during the fall, this data is for American larch, which is the deciduous evergreen species, deciduous conifer species. So for the fall, we can see that there are some differences between the amount of cold hardness that species can get, being the ones that are exposed to warmer temperatures, the ones without with less cold hardy. Winter, no differences, and spring, there is a high difference, meaning that there is a high chance of frost damage during the spring. If we look at the data of the evergreen conifer, we see that there is not really a pattern at all. And if we look in detail, we don't see any effect fall, winter, and spring. And if there's like one takeaway that I want you to have from this presentation is that the amount of susceptibility that species have differ, and in this case, deciduous conifers are the ones that are going to experience a higher susceptibility to climate change. Thank you.
Great, thank you, Francisco. So thank you to everyone who participated in the lightning talk competition. Let's give them one more big round of applause together. Please watch the screen and vote via the QR code for your favorite lightning talk presenter. The voting will be live for the next hour over the break. At this time, we invite you to grab lunch, stretch, and encourage you to engage with all of our presenters at the poster session outside. You might even see Bucky the Badger in the atrium taking in all of the posters. I really enjoy just seeing all the different kinds of plant science back to back. I definitely get stuck in my own lane, so it's nice to see that. This year, we are doing a poster competition as well, which will be running through lunch. Our poster presenters, both graduate and undergraduate, will be standing by their posters through the lunch hour. We encourage you to chat with them about their poster, and once you've had the opportunity to see all of the posters, please vote on your top favorite undergraduate and graduate posters. QR codes are located in the top corner of the poster boards. You only need to scan it once. And there will be a question of whether you are voting on the graduate posters or the undergraduate posters. We have 3 undergraduate posters, and their little piece of paper in the corner of their board will tell you that they are an undergraduate. And so please note that the QR code for the poster voting is different from this QR code for the lightning talk voting. And so our wall is opening up back there behind you guys. And so feel free to head out, get some lunch, and we'll meet back here at 1:30 for the second half of our symposium, starting with Stephen Schwartz. We'll see you shortly. Thanks.